This video is going to be on the Gram stain. The Gram stain is a differential stain and is used for microbial identification. I recommend that you view the other microbial identification technique video series and then also take the time to make note of the Gram stain. So as I mentioned, the Gram stain is a differential stain, meaning that it's going to use two or more dyes. In addition, this is going to be used to identify only bacteria. As we go through the Gram stain, I want to go through the steps and also talk about what a bacterium would look like after each step. The first question I want to pose is, what are the two results of the Gram stain? If you said Gram positive and Gram negative, that is correct. So as we go through each step, I want us to look at a bacterium that would be Gram positive or Gram negative, and after each step say, is it purple in color or red in color? In addition, I want to know how many steps are there to the Gram stain. If you said there are 4, that is correct. So there are 4 main steps to the Gram stain. The first step is going to be called the primary stain. The primary stain is going to be where we use crystal violet. Is crystal violet a basic dye or an acidic dye? Crystal violet is a basic dye, meaning it has a positive charge. This is important because the cell wall of a bacterium is negatively charged, so the basic dye will stain the microbe. So after this step, what color are our bacteria? The Gram positive is going to be purple, and the Gram negative are also going to be purple. I apologize, but I don't have a purple pen, so the blue is going to represent purple, and then red will represent red. After applying the primary stain, or the crystal violet, we're going to have the mordant step, specifically iodine. This is going to allow something to occur. This is going to allow the crystal violet to interact with the iodine to form large complexes. Be careful on how you word what is occurring in this step. Many students want to believe that this is helping the dye stick to the microbe, but it is not actually doing this. Rather, it is forming large complexes so that it cannot escape. So in this step, we're going to say the mordant iodine forms large complexes with the crystal violet within the peptidoglycan cell wall. This is a key phrase. If you're ever asked to explain what is occurring in this step, if you mention this phrase, you are going to receive the points you need. And the reason being is because it is talking about what is occurring and avoids the word sticks. And so you're going to be able to get all the points you need during this step. So now, what color is a Gram positive bacteria and what color is Gram negative? The Gram positive is still going to be purple, and so is the Gram negative. Because nothing has changed the color, we just added something to form large complexes. The next step is going to be the decolorizer, which is alcohol. In this step, we're going to only do it for 10 seconds. And that is because if we do it too much, we're going to decolorize both gram-negative and gram-positive. In addition, the alcohol is going to disrupt the outer membrane. Only gram-negative bacteria have an outer membrane, and so this is the only thing that's going to affect gram-negative. So we're going to say it disrupts the outer membrane of gram-negative bacteria. The third thing that's going to happen is going to depend on whether it's gram-positive or gram-negative. So when we look at a gram-positive bacteria, does it have a thick peptidoglycan cell wall or a thin? If you said it's thick, that's correct. Therefore, the gram-negative has a thin peptidoglycan cell wall. An analogy I'm going to use here is if we're going to take two sponges. We're going to have a thick sponge and we're going to have a thin sponge. So let's say that we take those sponges and we soak them in purple dye. Then we run them under the sink, and we run it just long enough that the thin sponge is going to lose all its color. So as soon as you see no more color being rinsed out, you pull them away. Because of the thick sponge, it's not going to have lost all its color. And this is exactly what is happening here. You're rinsing it just long enough that the thin sponge, the thin peptidoglycan cell wall, will lose its color, while the thick sponge, the thick peptidoglycan cell wall, is going to retain that color. And so depending on what you're describing, a gram-positive bacteria is going to retain the crystal violet because of the thick peptidoglycan cell wall, and gram-negative will not because of the thin peptidoglycan cell wall. I'm going to say gram-positive retains crystal violet iodine complexes because of its thick peptidoglycan cell wall. And the key word here is thick. While the gram-negative bacterium will not retain the crystal violet iodine complexes because of its thin peptidoglycan cell wall. Keyword is thin. So, if you're on the test and they ask you to do the gram stain for a gram-positive bacterium, I would make mention just that the gram-positive retains crystal violet because of its thick peptidoglycan cell wall and end the sentence there. If I was asked to just talk about a gram-negative, I would just mention the second half of the sentence. If I was just asked to describe the gram stain process in general, I would include the whole sentence. you probably could include the whole sentence and not have any issues because both ways are correct. It's just more information that you'd have to write down, and so it might be more simple and more targeted if you're just using the portion that applies to your bacterium that you're staining. So know, at this point, what is the color of the gram-positive and what is the color of the gram-negative? The gram-positive is still purple, again because of its thick peptidoglycan cell wall, while the gram-negative is neither purple nor red. Rather, it's going to be what we call translucent. Translucent means that there is no color. However, translucent is just the more technical term that we use in biology. So I would encourage you to memorize the word translucent versus colorless or clear. The last step of the gram stain is going to be the counter stain. And in the counter stain, you're going to apply safranin. Safranin is also a basic dye. If safranin is a basic dye, does it have a positive or negative charge? It has a positive charge because, again, it needs to stain the microbe and the cell wall is negatively charged. Therefore, now at step 4, the gram-positive is going to still remain purple, while the gram-negative is going to be red in color. So here, the reason why the gram-positive stays purple is because it retained the crystal violet throughout the first 3 steps. So although it may absorb some safranin because some was rinsed out during this step, the purple, the crystal violet, is a darker color and is more dominant, so no red is going to show through. While the gram-negative lost all its color during the 3rd step, and so therefore when you apply the safranin, it is going to completely absorb the safranin and it's going to stay a red color. You look at these results under the microscope. So you do the whole process and then you'll look under the microscope and see results. I would encourage you as well to take time to think about what would happen if you skipped one of these steps in the gram stain. For example, if I skip the decolorizer step, what would be the end result of my gram-positive bacterium and my gram-negative bacterium? If I skip this step, the gram-positive bacterium and gram-negative bacterium would still retain the crystal violet. So when you apply safranin, neither one would absorb the safranin, so therefore they would still be purple in color at the end. So I would just take a moment for you just to perhaps pause the video, or after writing this all down, to take a couple minutes and say, after each step, what would happen, or if I switch steps, what would be the end result? So this again is an identification technique that is used for bacteria only, and this is a differential stain. So I would encourage you again to view the other microbial identification techniques, but this is a very important technique that we use throughout microbiology.